Hi, my name is Christina.
My name is Zaf.
And welcome to our webinar. Today we are going to be presenting on the enzyme called maltase. Okay, so first things first, where is maltase found? It is found in humans, yeast, bacteria, and plants. But we're going to focus in this webinar on the role that maltase plays in humans. Okay, so maltase is a digestive enzyme. So this is a quick overview slide of digestion. As we know, digestion begins in the mouth with the mixing of food, and there's some breakdown, and then as it goes down the esophagus, that's the movement of food down the digestive tract, and then we have more of the chemical absorption or breakdown of food molecules happening. So that's where maltase is a key player. Maltase is involved in the chemical breakdown of food. Okay, so it is a last-ditch effort for chemical digestion. It's actually secreted by the brush border of the small intestine. So that's what this picture is here. It looks like a sea anemone, but it's actually the brush border of the small intestine. And this is where maltase is doing its work, breaking down disaccharide before absorption. Okay, so here's a simple mechanism of what maltase is up to. So this is a representation of our maltase enzyme. Here's maltose sugar. It's a disaccharide. So it comes in to the active site there of the maltase, and then it is cleaved, and then we get two glucose molecules leaving the enzyme. And actually, maltase, a single enzyme, breaks down over 1,000 maltose sugars into glucose in 1 second. So that one enzyme is doing a lot all the time. So yeah, we've got the two glucose molecules leaving, which are either absorbed or stored as glycogen in the liver.
So a few facts about maltase. The experimental PI was found to be 5.3, extinction coefficient 18.1, molecular weight 116 kilodaltons, and your pH is a little acidic, around 6.
Okay, so the genes that give rise to maltase, it is encoded by a gene called MGAM. That gene is about 82,000 base pairs long, and it's located at chromosome 7q34. The enzyme consists of 1,857 amino acids, but we're going to be focusing on the key players, the amino acids at the active site. It shows structural similarity with the intestinal enzyme sucrose isomaltase. Both sucrose isomaltase and maltase have the same two catalytic sites. All right, so this is another slide that's kind of talking about the relationship between maltase and sucrose isomaltase. We said they're close cousins because they're members of the same family, the glycosyl hydrolase family, 31. They have identical 48 exon structures, and they actually work together in the intestines to break down sugars. So maltase can break down 1,4 glycosidic linkages. Sucrose isomaltase can break down both 1,6 and 1,4 glycosidic linkages. And then they have shared peptide domains. So here we've got the predicted tertiary structure for both MGA, which is maltase, and SI, sucroisomaltase, described as, then we've got the beta and alpha helices, and that's the repeated pattern throughout the enzyme. So all of this together, the shared exon structures, peptide domains, it suggests that maltase and sucrose isomaltase evolved by duplication of the same ancestral gene. So they're closely related, working together. Okay, kind of shifting gears here. Before we introduce our article and our study that we looked at for this webinar, we're going to bring some facts from the Center for Disease Control and Prevention because it relates to why our researchers did the study that they did. So. As we all know, diabetes and obesity are prevalent in the United States, and basically this is all just information showing that the CDC says that those rates are continuing to increase. Newly diagnosed type 1 and type 2 diabetes are on the rise among children, teens, and adults. Diabetes decreases the quality of life, shortens life expectancy, and increases healthcare costs. Over 12% of adults are estimated to have diabetes, and 1/3 of people over the age of 20 are pre-diabetic. And then with obesity, nearly 40% of Americans are considered to be obese, and that percentage has been increasing over the years. And then obesity-related conditions, it increases your probability of stroke, heart disease, or certain types of cancer. So our article is trying to figure out how we can combat this issue. Okay, so this is article, it was from the Journal of Physical Chemistry. These researchers looked at the mechanistic pathway on human alpha-glucosidase maltase, glucoamylase, unveiled by quantum mechanic and molecular mechanic calculations. So basically they are trying to look at the exact pathway that maltase uses in order to inhibit it, find a competitive inhibitor so that they can produce therapeutical agents for people who are predisposed to obesity or diabetes, and it's inhibiting or delaying the breakdown of sugars for them.
All right, here is the 3D picture of maltase. You can see all 1,800 amino acids and the beta barrels, alpha sheets, alpha helices. So from the methods of our primary literature, they use quantum mechanic and molecular mechanic calculations and molecular dynamics to map the active site of maltase. What they wanted to do was see which amino acids play a critical role in the action of maltase, so which ones are actually binding to the substrate, and how that active site looks and what they can use to mimic a substrate and prevent the absorption of sugars. So they used onium, which is our own n-layered integrated molecular orbital and molecular mechanics device to find the angle distances between molecules, which help them determine the geometry and the distances between the amino acids to better map what would be a better substrate to fit in to inhibit the enzyme action. And in that same vein, they used the density functional theory and a hybrid functional to create a map of what that area looks like in those amino acids. So this is a little more detailed mechanism of what maltase does. This is your enzyme maltose molecule that undergoes the transition state, where it's cleaved and then it leaves as 2 glucose molecules. So as you can see here, this follows they propose that it follows the Koshland double displacement mechanism. And so in that you have an acid that donates its hydrogen right here at oxygen 1, where your glucose 1 and glucose 2 molecules are bound. That hydrogen bonds with this oxygen and it goes away to glucose 2, forms an alcohol there. And a nucleophile comes and attacks, and it attacks at carbon 1, and it forms a bond there with the oxygen. This is your glycosylation step. Then, while your nucleophile is still attached, to go to the deglycosylation step, you have water coming in, and it reacts with that carbon-oxygen bond to break that and leave your glucose 1 with that alcohol as well. So at the end, you're left with 2 identical glucose molecules with alcohols on them. So in our paper, they looked at this, and that acid is aspartic acid 542. It comes in and donates its hydrogen. There's your oxygen right there. This is what they mapped out. And then it undergoes the transition state, where you can see your aspartic acid 542 here and the oxygen, and as well as your aspartic acid 443, which is the nucleophile, and that's attached to your glucose 1 molecule, and it forms your first intermediate in the glycosylation step. Going into the deglycosylation step, you have aspartic acid 542 here, and water has come in, and it reacts with your aspartic acid 443 at the nucleophile, and it undergoes the transition state. Then your products are formed with 2 identical glucose molecules.
Okay, this is the energy pathway of that whole reaction that Zaf just explained. So here we've got the reactants, transition state 1, intermediate 2, transition state 2, and products. So as we can see from this, the rate limiting step of this reaction for maltase is that first intermediate, that first transition state, because the energy is higher than the transition state 2. So that's the rate limiting step, is that glycosylation. Okay, so for the results, as Zaf mentioned earlier, the reaction follows the classical Koshland double displacement mechanistic pathway, where you have that attack at the anomeric carbon and then the acid coming in, forming the glycosylation and deglycosylation steps. And they found that there's high inhibitor selectivity for maltase, so this is specifically at the N-terminus, that's why they have NT there. And then, so basically what that means is that they can't just find like a general inhibitor to inhibit the maltase pathway to delay the breakdown of sugars. They need something that is really specific and that's going to copycat maltose pretty precisely if they're trying to, decrease the rates of diabetes and obesity. So, and then for the high layer, this is what it includes. These are all the amino acids they found, particularly, as Zaf mentioned, the key players for the high layer are the aspartic 542 and the aspartic 443, which are acting as the acids.
So we went a little bit into the pharmacology of what they can use to inhibit the maltase mechanism, and basically they were looking into this and they found that the inhibition of the enzyme activity, specifically at the N-terminus, it's going to inhibit the enzyme action, which is going to delay the delivery of sugars into your digestive system. And originally they were using acarbose for this, but they found in recent research that catalinol is 300 times better at inhibiting this particular enzyme.
Okay, so now we're gonna shift gears and talk about what happens when we don't have enough maltase. So no longer talking about inhibiting it, but what happens when somebody's born without enough. So there's a disease called POMC disease or POMC deficiency. It's an autosomal recessive disease characterized by a buildup of complex sugars in the body. So you don't have maltase, you're not breaking down those maltose disaccharide sugars, so you have a buildup. There's different ages of onset: classic infantile, non-classic, and late onset. So some people are born with it, so it develops some months after birth sometimes, or later into the teenage years. The symptoms of POMC disease are weak muscles, poor muscle tone, enlarged liver and heart, inability to gain weight, trouble breathing, problems with respiratory system, and high levels of creatine kinase. So it's not a disease that you can kind of just live with without any treatment. So in order to treat people with maltase deficiencies, doctors do an enzyme replacement therapy. So a genetically engineered drug is given intravenously that acts like naturally occurring maltase, essentially. Acid alpha glucosidase enzyme, which is maltase, another word for describing maltase. So thank you for listening. This is how we put together this webinar for you. We used all these different references and research papers. Thank you. Here's an ending cartoon of competitive inhibition for you. Please comment below with any questions you might have for us.